Positive selection coordinates the expression of CD4 or CD8 with the specificity of the T-cell receptor and the potential effector functions of the cell. At the time of positive selection, the thymocyte expresses both CD4 and CD8 co-receptor molecules. At the end of the selection process, mature thymocytes ready for export to the periphery express only one of these co-receptors. Moreover, almost all mature T cells that express CD4 have receptors that recognize peptides bound to self-MHC class II molecules and are programmed to become cytokine-secreting cells. In contrast, most of the cells that express CD8 have receptors that recognize peptides bound to self-MHC class I molecules and are programmed to become cytotoxic effector cells. Thus, positive selection also determines the cell surface phenotype and functional potential of the mature T cell. Selecting the appropriate co-receptor for efficient antigen recognition and the appropriate program for the T cell's eventual functional differentiation and an immune response. Experiments with mice made transgenic or rearranged T cell receptor genes show clearly that it is the specificity of the T cell receptor for self-MHC molecules that determines which co-receptor a mature T cell will express. If the T cell receptor transgenes encode a receptor specific for antigen presented by self-MHC class I molecules, mature T cells that express the transgenic receptor are CD8 T cells. Similarly, in mice made transgenic for a receptor that recognizes antigen with self-MHC class II molecules, mature T cells that express the transgenic receptor are CD4 T cells. The importance of MHC molecules in this selection is illustrated by the class of human immunodeficiency diseases known as Bare lymphocyte syndromes, which are caused by mutations that lead to an absence of MHC molecules on lymphocytes and thymic epithelial cells. People who lack MHC class II molecules have CD8 T cells, but only a few highly abnormal CD4 T cells. A similar result has been obtained in mice in which MHC class II expression has been eliminated by targeted gene disruption. And that's positive selection coordinates the expression of CD4 or CD8 with the specificity of the T cell receptor and the potential effector functions of the cell. Have a great day.